Y'all, thank you so much to everybody joining us in person and on Zoom for this. I don't keep track of the numbers, but this is certainly our first in-person Applied Ecology Minors Research Symposium in 5 semesters. It's great to be back. I'm thrilled to not be sitting in a chair at home leading these in a webcast, though they were still very useful to join remotely in those times. First, I want to remind you that we have 2 symposia. So today you'll be hearing from 4 speakers, and on Friday at noon, you'll be hearing from 4 more speakers. So please continue to join us on Friday in person or on Zoom as you are able. Maybe. All right. Today's lineup, our presenters are Lucie Ciccone, Paige Mesecar, Margaret Haney, and Mary Grace Ussary. So before we get started, I want to acknowledge that we are on the traditional lands of the Skarura Tuscarora, the Catawba, and the Lumbee tribes. The goals of applied ecology are to study the natural non-human world around us and our human interactions with the world. Often we can apply what we learn to better manage or conserve natural resources, but it's important to note that way before Western scientists decided that this was important, expert knowledge of the natural world was already applied sustainably as an integral part of indigenous cultures. We are privileged to be able to practice science and to study ecology in academia. But I hope that we can remember that the Western, the scientific, the academic way is not the only way, and it is likely not even the best way. Acknowledgement is a form of decolonization or dismantling an existing power structure. In order to change systems of oppression, we need to first acknowledge that the system is built on oppression. North Carolina State University is a land-grant university, which means it originally received funding through the Morrill Act. That's federal legislation signed by President Lincoln that bought or forcibly took land from indigenous tribes across the United States, including in North Carolina. Indigenous lands raised $134,920 to fund NC State. That's the equivalent of $3,810,578 today. I hope we can disrupt and dismantle colonialism beyond a simple land acknowledgement by supporting and empowering each of you as students in our classrooms, in our research programs, and beyond. So, without further ado, I am going to close this presentation, open Lucie's presentation, so bear with us for a little bit of technological transition, and away we will go. Yeah,
thank you.
Mm-hmm.
Hi, my name is Lucie Ciccone, and today I'm going to be presenting on work that I did under the mentorship of Dr. Elsa Youngstedt and Michelle Kirchner, investigating the comparative thermal tolerance of needle ants, which are an invasive ant species present in the southeastern United States, nesting in both the canopy and the ground. I want to start by saying that invasive species are known to pose threats to biodiversity and ecosystem function in their introduced ranges. And in managing invasive species and developing conservation strategies, it's increasingly important that we take into account how invasive species are responding to climate change and the role that thermal ecology might play in shaping their or affecting their spread. And so in light of this need, in this project, I looked at one invasive ant species through the lens of thermal tolerance. So this is a needle ant, Brachyponera chinensis. They're a small insectivorous ant species introduced to the Southeast United States from Eastern Asia in the early 1900s. And since then, they have been found in states throughout the Southeast and Eastern US, both in disturbed forest habitats and undisturbed forests, which is something different from other invasive ant species here, which are mostly just found in disturbed areas. Understanding where they're expanding and what their range is important ecologically. Apologies for the quality of this photo. This is a nice picture of some native seed dispersal ants, which have been known to be to decrease in abundance in areas where needle ants have been found to be introduced. And this seed dispersal is a really important ecosystem function that might be impacted by needle ant expansion. But needle ants also have direct concerns for human health. Like fire ants, they have a venomous sting that can induce anaphylaxis. But despite the ecological and health significance of this species, we don't fully understand what the mechanisms are that give this species a competitive advantage. There have been some previous studies looking into the role that cold tolerance might play in facilitating needle ant spread. But these have been somewhat inconclusive since they haven't consistently found that needle ants have a higher cold tolerance than the species that they displace. And so this indicates that cold tolerance might not be a particularly driving factor explaining needle ant range expansion. But the heat tolerance of needle ants remains to be explored. And so last summer, during field work, Michelle, who I work with, found needle ants nesting in the canopies of trees in North Carolina. And this was really cool and interesting because previously needle ants were just thought to nest on the ground. Finding these ants in the canopy raises new questions and opportunities for understanding needle ant thermal ecology, because we know that canopy habitats get both hotter during the day and experience more variable temperatures than habitat on the ground. So it's possible that needle ant heat tolerance could help explain not just their presence in the canopy, but also throughout the Southeast. So building from this, we asked, does heat tolerance facilitate or otherwise shape needle ant presence in the canopy? And hypothesized that canopy ants would have adapted or acclimated to a hotter and more variable canopy environment. I've been using some terms related to thermal tolerance and wanted to pause to talk about what we meant during this study. So we chose to look at the extremes of what needle ants were able to survive. So we looked at critical thermal maxima, or the maximum temperature at which these ants were able to continue to function, and then their critical thermal minimum, which is the lowest temperature at which that's the case. And so I'll be referring to CT max and CT min, and that just sort of refers to this range of temperatures in which needle ants are able to survive. And so with this in mind, we predicted that needle ants that were nesting in the canopy would be found to exhibit a higher critical thermal maximum than needle ants nesting on the ground, but that these two groups would have relatively similar critical thermal minimum values. And so to answer this, we collected data as part of a larger research study carried out by Michelle and designed by her during the summer of 2022. We worked at 4 sites across central North Carolina and collected ants from 12 trees at these sites. At each tree, we located both ground nests. So on the left, these flags on the ground are where we've located ant nests in the ground. And then on the right, this is Michelle climbing a tree and to locate nests in the tree canopy. And then once we had located these nests, we collected ants to bring back to the lab, which is what I'm doing in this center photo. We also collected temperature data from nests at each tree using thermocouple loggers, which is what's present here in the forefront of this picture on the left, which we stuck into the entrance of nests on the ground and in the canopy. And so in the lab, on the same day as collection, we ran thermal assays on the ants that we collected. We ran assays on 10 individuals from each nest, with 5 ants being used to test CT maximum and 5 for CT min. And these assays involved placing individual ants in vials in a heating block, letting them acclimate, and then incrementally raising or lowering the temperature by 1 degree Celsius every 5 minutes. And so then at each interval, we would check on the ants and then record the temperature at which they were no longer able to move. And we were ultimately able to get needle ants from 38 nests, with 9 of these coming from canopy nests and the remaining 29 coming from the ground. So there was a difference in sample size between these groups, which I'll get back to later in the presentation. Now, a goal of mine with this project was to get more experience with the data cleaning and analysis part of ecological research. So I've been really lucky and grateful to be able to participate in the fieldwork components of this project. And this semester, I was able to learn a lot about what goes on after that, once the data has been collected. So I wanted to talk a little bit about what these data cleaning and then analysis steps looked like. So first, we did a lot of data organization. We calculated some average values that we were then able to use in our models. So we calculated average CT max and min by nest. And then we created graphs of our raw data to understand the spread and shape of our data sets. And we use this to identify an outlier in our data. So that's what's shown here in this Cleveland dot plot. And we were able to exclude this in our analysis. Then for the analysis itself, we tested our hypothesis using a linear mixed model in which we treated stratum, so ground or canopy, as our explanatory variable, and included site and tree as nested random effects. We ran this model separately for our 3 different response variables of interest, so critical thermal maximum, critical thermal minimum, and then we also looked at the difference between these values, which we called thermal breadth. And then to check our model results, we analyzed model residuals and ran an analysis of variance. And so what we found was that there was a marginally significant difference in critical thermal maximum values between ants nesting in the canopy and on the ground. Looking at this box plot here, this is actually a modified box plot where the midlines are the estimated marginal means for CT max for canopy ants on the left, and then ground nesting ants on the right. And then the bounds of the box reflect the standard error, and then the tails are the confidence intervals. And so while there was this significant difference, the actual difference in estimated average CT max values they differed by less than ½ degree Celsius between these 2 groups. And so it's likely that this difference, while potentially significant, is not biologically very relevant. But the presence of there being at least a slight difference did follow our initial prediction. We also did not find any significant difference in average CT minimum values between these 2 strata, which was also in line with what we originally thought. Now, to contextualize this data for what this means for ants and what they experience, we also need to take into account the temperatures that these ants are experiencing and living in. And so to do this, we used the thermocouple data from the nests and looked at the daily maximum temperatures in the canopy and ground nests. And so that's what's shown here. These are this is another modified box plot. And these are the daily maximum nest temperatures for nests in the canopy on the left and then on the ground in the right. And as expected, we did find that nests in the canopy had daily maximum temperatures that were higher than those on the ground by about 3 degrees on average. But while this is a considerable difference in experienced temperatures, all of these temperatures were still below needle ant critical thermal maximum that we measured. So this red line is CT max for needle ants. And so there were never days where the maximum temperature exceeded that CT max. So while there was still this difference and temperatures did not exceed the critical thermal maximum, looking at the difference between maximum daily temperatures experienced and CT max can offer insight into the vulnerability of these 2 groups of needle ants to climate pressures. And so this value is known as warming tolerance, and can be thought of as a quantification of sort of the amount of climate buffer that needle ants living in the canopy and on the ground have before experiencing temperatures that are fatal. So the larger an ant's warming tolerance, the less likely they are to experience a temperature that might be fatal. And so when we compared these warming tolerance between the two strata, we found that there was a significant difference between canopy nesting and ground nesting needle ants, with ants collected from the canopy having a significantly smaller warming tolerance than those found on the ground. And so additionally, while both groups of ants were found to have warming tolerances that were pretty large, so here what this means is that the average warming tolerance for canopy ants was about 12 degrees Celsius. That's sort of the amount of degree buffer that they have, which is pretty large. What we can see is that there were days where there were temperatures that got within 5 degrees of the temperature at which needle ants would die, which is, as climate change occurs and temperatures increase, we might expect that the number of days where this happens could increase. So taken together, our predictions of different thermal maxima was only tenuously supported, indicating that needle ants in the canopy have not adapted or acclimated their heat tolerance to the warmer canopy environment. But this lack of biologically significant difference in thermal maxima, coupled with the significant difference in experienced temperatures, could indicate that canopy nests will be more climatically limited than ground ants, since these ants will be more vulnerable to fatal temperatures under warming climate scenarios, when the number of days with maximum temperatures close to needle ants' critical thermal maxima will increase. I also want to reiterate that the lack of arboreal data that we use makes inference hard, especially considering the sort of inconclusive significance that we found with critical thermal maximum. And so it's possible that with more arboreal data, we could have found a more definitive difference between these groups, or that they would have been more clearly the same. Moving forward, there are several questions that I'm interested in with needle ants. Some of these hopefully will be answered, or we will get more information from them through Michelle's research, including how needle ant thermal tolerance varies with season and how it compares with other species present in the canopy. And then another important question is understanding what needle ants' thermal optimum is. So we looked at the temperatures that they're able to survive at, but not the temperature at which they might thrive the best. And so understanding that can also help explain where they might go and what habitat they might utilize and why that's the case. So I want to give a really tremendous thank you to Elsa and Michelle for their mentorship throughout this project, and I would be happy to take any questions.
It's quite interesting. Great job, I like. It's quite interesting that these ants are both successful in disturbed and natural habitats, right? I think you mentioned that that's kind of the rarity for exotic. What can your findings of arboreal ground nesting tell you, or predictions of the urban gradient and kind of the higher temperatures in disturbed human habitats that have, a lot of impervious surface and the heat island effect? And maybe you guys are already kind of working on that aspect of it, but would you have any predictions based on your findings here?
I haven't thought about that question specifically. I think it is interesting. I know in their native range, they're commonly found in urban settings. And so there's been research done on needle ants in and around like street green spaces. It was interesting that we didn't find differences in their heat tolerance. So in that way, they might be less able to persist in urban areas with the increasing heat island effect. But in the meantime, before their sort of critical thermal maximum is reached, it's also possible that there's something about being in a warmer, like canopy environment that is advantageous to them, sort of getting to the concept of thermal optimum. And so there might be other factors of like urban spaces that they're able to utilize. I think it also would be interesting to think about and do more investigation of like how they're interacting with the different species, both on the ground and in the canopy. And because these species differ, there might be different mechanisms explaining their presence in those two areas. And then Michelle's research also is looking at differences in ant species across that urban gradient. And so they'll be interacting with different species too. So there's a lot of factors to consider there. And I don't have a prediction at this moment, but definitely interesting to think about.
So you have some chat icons.
Oh, thank you. I'm
Yeah,
sorry.
no. It went hiding up at the top.
Erin, do we have time for another question? Because
I
Jim's
think
got
we
that
have-
one in the chat.
Yeah, we've got time for one more. Jim is asking, do you think there might be a bigger temperature difference if measured down in the nest than just at the nest entrance? And any idea what the thermal tolerance of larvae are?
Second part of that question, I do not. The first part, probably. These nests can be pretty extensive. So I guess like especially ground nests, there's probably going to be a larger temperature difference if, well, it's hard to say. I think there would be, we did try to, I should say, with our thermal couples, like get them within the nest itself. So not just like at the entrance to the nest, to account for that. But it would also, I don't know what the temperature difference is between like.
Maybe another way of putting the question is, do you think that the nests are like buffering or amplifying
Yeah.
canopy ground climate difference?
Probably buffering, given that, or amplify because the ones on the ground, like, probably. I would have to think about that. I think like soil and then tree matter in the tree respond differently. So how that buffer would exist would be different for tree nests as opposed to ground nests. But the extent of that buffer, I'm not sure. Yeah.
What are the physical properties of
Yeah.
wood compared to
Yeah.
soil? Awesome. Thank you so much. And thank you for your questions. Up next, we have Paige Mesecar. And let me go ahead and grab that menu.
Thank you.
Yeah, sure thing. And those were thank yous and great jobs in the chat for you. All right. You might have to click the screen to advance the first one, but then you should be ready to go.
All right, good morning. My name is Paige Mesecar, and I'm going to present on the effects of parasitism and diet on the common eastern bumblebees. All right. So the species at play here are, like I said, common eastern bumblebees. These have been extensively studied as models for insect decline. They are generalists, so they're not actually declining as much themselves, but they are good models. Crithidia bombi is the parasite that we used. This has been implicated in, like I said, insect decline. However, sunflowers have been shown to reduce the effects of Crithidia. A previous PhD student in Dr. Irwin's lab published a thesis showing that wild bumblebees with access to sunflowers have lower levels of Crithidia. So this current project is April Sharp's PhD thesis in Dr. Irwin's lab. We're trying to fill in the gap to expand on that research to figure out the exact, the specific degree that Crithidia and diet impact bumblebee performance. So we're asking, does sunflower pollen diet recuperate activity levels of bees infected with Cryptidia? Short answer, yes. Long answer, we measured the time that bees spent visiting flowers, the number and proportion of flowers visited, and the amount of pollen deposited on each flower. So methods. Excuse me. So to begin with, we infected or inoculated bumblebees with Cryptidia. This sounds pretty straightforward, but there's lots of steps even within that. We'd isolate the Cryptidia, get the right proportion, and then try to convince these bees to eat from a pipette that is frankly terrifying to them. Then the bees were isolated in these microcolonies for 1 week. Excuse me. And they were fed wildflower pollen this entire time. In the far corner, you can see the sucrose wells. That was their carb source. And then the pollen was fed in the top of test tube capsules. The wildflower pollen is a control meant to mimic what they're eating in the wild. It has a large variety. One of the possible issues with sunflower pollen is that there's lower protein and variety of nutrients. So after this point, 24 of the bees were switched to sunflower pollen as a treatment. And then 70 days after that diet change, 1 to 3 bees were introduced to a mesh cage, which I will get to in just a second. Here's a diagram of the treatment groups. There are 12 in each and 48 total. We did, I think, 18 groups throughout the summer, so a lot of bees. One of the major issues with this setup was the mortality rate. A lot of bees died from this, and so that created some issues trying to get significance in their data. With data collection, we did voice recordings and notes. This is our setup. There's a mesh cube with tomato plants and snapdragons inside. 1 to 3 bees were introduced for 30 minutes, and then we analyzed the behavior. We collected bee ID, plant ID, if the bee foraged, just yes or no, the amount of time in each flower, and then the number and proportion of flowers visited. Part of that too was collecting the individual flowers and then freezing them. These were then dissected, and we analyzed the number of pollen granules posited. And then the bees themselves were also dissected, and then the Cryptidia counted in each bee, which gave us this. So the uninoculated colonies showed 0 Cryptidia, which is good. Our methods worked. And then I forgot to write down the p-value, but there is significance difference in the sunflower pollen treatment versus the wildflower pollen. Should go back. So this is pretty expected. It's mostly just to confirm that there's no Cryptidia where we wanted to, and that there was a difference in the treatments. And then this is a simple linear regression. Unfortunately, it's not statistically significant, but the trend is in the direction that we expected. Cryptidia, or bees with lower Cryptidia, showed decreased activity. Foraging is an active thing. They're trying to gather pollen. Later, there's data on the amount of pollen distributed to the stigma, which is not like super intentional. They're trying to forage for food. The visits, this is the proportion of flowers visited, just on the snapdragon data. And again, it's not significant, but the trend is in the right direction. So hopefully further research would show the same thing, just significance. And then the pollen distributed, still not significant, but the trend is in the right direction. And discussion. So the decline in pollinators is definitely disturbing for a lot of ecosystem diversity and agricultural plants. Bumblebees specifically are important to tomatoes. They're one of the only species that can pollinate that well. The preliminary data shows promising trends, but this definitely needs to be replicated. April, I believe, is continuing this project, but without the microcolonies, which she thinks is the cause of the decline or the mortality. So they're going to use entire colonies, inoculate those, and see if that works better in terms of keeping the bees alive. That won't give as specific data in a couple ways. We measured the amount of sunflower pollen but that the bees ate by weight over the course of their treatment. I did not analyze that for this because it got very minute, but that is possible for her to include in her thesis. But hopefully that using the entire colonies as a treatment will work better to get more significant data. And then April Sharp and Dr. Rebecca Irwin were a huge help helping me learn all of this from scratch. We really jumped in day one. I was already dissecting bees right off the bat, so I learned a lot from them over the course of the summer. Any questions? Like the chat. Sure, yes.
It's really great that you're able to replicate what has been shown before to show that it's a robust and real effect. And that's
Right.
always really good to see. But I wondered if, any subsequent behavioral differences might be never being affected, infested with Graffitidia versus having it and then having gotten rid of it.
Right.
So, I'm wondering if there's been any discussion of an experimental design where you infect everybody
Mhm.
and then you like cure your uninfected group
Right.
and then kind of do the test, right? I just want to hear your thoughts on that. Do you guys discuss that at all, or do you think that that's just kind of a ludicrous thing to include?
I hear what you're saying. I did not write the protocol for this. I mostly just joined April on her thesis. I think one issue with that would be there would be no control. The previous PhD student only did, I believe, field work. So we were trying to find the exact degree with controls. And if we infected everything, we wouldn't know what the baseline is of like never ever having Cryptidia. Hopefully we got enough replicates of that of having been inoculated, but then given the sunflower pollen. But we'll need, like I said before, a much higher degree of that in order to show the actual effect and not just trends. Does that answer your question?
Mhm.
Okay, yes.
Speaking of the bees who were uninfected, it seems like for the zero Cryptidia bees, like there was a very wide range of activity.
Yes.
Used before, some of them did a lot. Do you
Mhm.
have any sense of what's going on with that? It's like that's the thing that's making your regression weird because that
Yeah.
baseline is just all over the place.
Mhm.
Are
There's
there other
definitely-
things
sorry.
that you might be able to like include or account for that tell you whether a low Cryptidia bee is gonna forage or not?
We did kind of change protocols throughout this summer. So at first we introduced one bee, trying to like really make sure we figured out what was going on, but they spent an entire 30 minutes trying to get out of the mesh cage and not actually foraging. So then we introduced more bees, so it was like 2 or 3 at a time. And they seemed calmer when they were like around their friends. That did a little better. And we also added sucrose to the flowers themselves, which also encouraged foraging. So since this is preliminary data and those changes happened, like there's definitely some issues with trying to get the bees to forage. Hopefully that's figured out, and then starting with the entire colonies can have like perfectly consistency. But that definitely affected my data.
So that data set you showed us and kind of follow the variation, like one bee, multiple bees. extra sucrose.
Yes,
Thank you.
And did you have a question in the chat?
I think so. Yes.
There are.
Awesome. So Jim is asking, the graph suggests that your activity data may not be linear. Maybe try analyzing with a nonlinear model. Have you looked at that?
I've not, no, this was my first attempt at R and data analysis. So we went pretty simple with this. Going forward, that would be a great thing to try. See if that makes more sense with the
Yeah.
data.
And do you know that there is not any sunflower pollen in the wild flower pollen mix?
No, I do not know the specific components of the wildflower pollen. Sunflower pollen might be included, but I think it would be to such a low degree that it wouldn't be additional.
A dilute effect, if any.
Right.
Awesome. Thank you, Jim. Thank you, Elsa and David. Thank you, Paige.
Thank you.
Up next, we have Margaret Haney. So I'll grab the chat again for you. All right, so that means you may have to click your first click on the screen to advance.
Okay.
If you use a trackpad.
Awesome.
All right.
Thank you.
Uh-huh.
All right. Hi, everyone. My name is Margaret. I'm a senior in environmental science and I'm part of the Applied Ecology program. And this summer I had the opportunity to work in the Bonanza Creek Long-Term Ecological Research Station in Fairbanks, Alaska, doing research on nutrient limitation in boreal streams. So I'm going to start off real quick with a quick introduction to Alaska's boreal forest and some challenges that it's facing in the face of climate change. The first challenge that we're going to talk about is wildfires. Wildfires are becoming more frequent and more intense due to a changing climate, like in many other regions of the world. And unfortunately, you know, this trend is only expected to continue into the future and become more intense. The second challenge is a little bit more unique to the region, and that is permafrost thaw. As we are seeing higher temperatures in Arctic areas, permafrost is starting to thaw. If you see in this diagram here, what permafrost is soil that's been frozen consecutively for 2 or more years. On top of permafrost is the active layer of soil that thaws and refreezes annually. So with the change in climate, the active layer is starting to deepen, which means that it's starting to intrude into that permafrost layer that's been historically frozen for sometimes tens or hundreds of thousands of years. So you can imagine that will have like a number of really extreme impacts on these ecosystems. Both of these processes release nutrients into their surrounding environment from trees and other kinds of vegetation burning that stores a lot of nutrients, which are then released available to the environment. And permafrost soils do hold a lot of nutrients as well. So when those are frozen and then thaw, they'll release nutrients that are not typically available to that environment. And then a lot of these nutrients can end up in streams since they're a receiving ecosystem. This is really important because of how it influences and affects biofilms, or streams' primary producers. Biofilms are like the conglomeration of algae, bacteria, and fungi that are found in these ecosystems. These biofilms contribute to a number of really important ecosystem processes that streams are a part of. For example, they are primary producers, so they are very low on the trophic pyramid, and they support the food and energy needs of higher trophic levels. They are also photosynthesizers, so they are a big influence on the difference between the amount of carbon dioxide that's released and the amount of carbon dioxide that's stored through photosynthesis and respiration. And then that in turn can influence to what degree a stream is a carbon source or a carbon sink. Biofilms also take up nutrients from their surroundings, which is why they are so sensitive to changes in nutrient concentrations in their streams. Most streams are nutrient limited, which means that they don't have high enough nutrient concentrations to support larger biofilm communities than what is already present. So that means with an addition of new nutrients from things like wildfires and permafrost thaw, we could see a big change in the in biofilm population sizes. Therefore, when I went to this research station, my question that I was there to answer was, are primary producer populations in interior Alaska's boreal streams nutrient limited? And if so, by what? Previous research done on this topic has suggested that boreal streams in other regions are definitely nutrient limited, and mostly by nitrogen. So I decided to extrapolate that to Alaska's boreal streams. And this was my hypothesis. So in order to determine whether or not a stream is nutrient limited, what we can do is add trace amounts of these nutrients into the stream and then measure the impact that that has on biofilm populations. So if you were to add nutrients to a stream that is nutrient limited, you will see an increase in primary producer populations because they have access to all of these nutrients that they were needing. If you add nutrients to a stream that's not nutrient limited, it has enough nutrients already, so you will most likely not see a significant change in primary producer populations. So in order to make this happen in real life, I did a nutrient limitation study with nutrient diffusing substrata. It's these little blue cups that you see up here. They're composed of agar that's mixed with different nutrients. And then I put a little glass disc on top of all of them that allows biofilms to colonize on the disc while also being able to access that nutrient that's available underneath. So I made 4 different batches of these NDS cups. I had a control, I had one that had nitrogen added, one with phosphorus, and one with both nitrogen and phosphorus. I put these cups in 9 different streams, let them sit there for 27 to 31 days to allow like algae to colonize on it. And then I removed the cups. That's what they'll look like when they come out of the stream. I extracted the chlorophyll A from the glass discs and ran it through a spectrometer and did some math to figure out how much chlorophyll A was present on these discs. The reason that I did that was because it's a really good proxy for primary producer biomass, since they're all photosynthesizers, they all have chlorophyll A. And so that is one of the most convenient ways to measure primary producer biomass. I sampled 9 different streams. We selected different sites that had a variety of permafrost extent conditions and a variety of years since the most recent burn in that stream's catchment. Permafrost extent is basically like how continuous permafrost is in that area. So areas with low permafrost extent, the permafrost is very patchy and discontinuous, while in high permafrost extent areas, it's very consistent throughout the whole region. So just a little bit about these sites. The year in parentheses under the burn sites are the year of the most recent major burn. We had 2 sites that were burned recently and had low permafrost extent, 4 that were burned with high permafrost extent, 2 that were unburned with low permafrost extent, and only 1 that was unburned with high permafrost extent. It's becoming increasingly more difficult to find sites that are unburned. So that's why the majority of our sites were burned. So now into results. There were 4 different kinds of nutrient limitation that we were able to observe in the study. I'm not going to show every single graph for every single site because like after 9 sites, it gets a little bit overwhelming and kind of repetitive. So what I'm going to do is talk through each of the 4 different kinds of nutrient limitation, just so we get a good understanding of like what was going on in these streams. I used an ANOVA to analyze the difference between means of each treatment and then Tukey's test to examine how these means differed from each other. So this is the first example of a site. This is Caribou Creek. You see on the x-axis, we have each treatment. The y-axis shows the amount of chlorophyll A that was present and therefore like the amount of biomass that was able to grow on each treatment. This stream was primarily phosphorus and secondarily nitrogen limited, which means that phosphorus was responsible for the majority of nutrient limitation occurring in that stream. But when you added nitrogen to the phosphorus, it was a little bit more limited. You can see the differences in means between these four box plots and the fact that phosphorus was significantly different, but nitrogen and phosphorus together was even more significantly different, shows us that it's primarily phosphorus and secondarily nitrogen limited. 2 out of the 9 streams were limited in this way. And then next was Moose Creek. This was co-limited, which means that nitrogen and phosphorus kind of had an equal amount of responsibility for the nutrient limitation in the stream. 2 out of the 9 sites were also co-limited. Shovel Creek was the only phosphorus limited creek or stream, which means that phosphorus was the sole reason for nutrient limitation in that stream. And then the remaining streams were not limited at all. So you can see there's no significant difference between any of the treatments. What's most important here is that 5 out of our 9 streams were nutrient limited, which means that the majority of the streams that we sampled in this region were nutrient limited. It's also interesting to note that none of them were limited by nitrogen alone, which is what we predicted was going to happen because that's how it is in other boreal forests. So that's kind of the mystery. Alaska has had really high background nitrogen concentrations and we're not really sure why. But yeah, very interesting there. Some pitfalls to my study. I lost many NDS cups to streams, unfortunately. We didn't really tie them in as sturdy as we should have initially. This is a complete bar that we are able to retrieve, but most of the times you would see like every 4 or 5 cups, 1 or 2 would be missing, which is really unfortunate for data analysis. It was really sad. But if I were to do this again, making sure to secure the zip ties a little bit more would be the way to go. Also, the small number of sites that we studied means that we couldn't really find any relationships between the characteristics of the streams, like how recently the last burn has been, and the permafrost extent of these sites. We weren't really able to find any trends between that and nutrient limitation in those streams. So what we were able to take away from this research is that the majority of streams in Alaska's boreal forest were nutrient limited. So after receiving influxes of nutrients from disturbances like wildfires and permafrost thaw, we are likely to see an increase in primary producer populations. This information is hopefully going to be helpful in further research concerning resiliency in the face of climate change. Also, boreal stream productivity and a stream's ability to support primary producers and higher trophic level organisms. And finally, the contributions of streams to greenhouse gas release. So this is all work that other members of my lab are conducting now. The LTER is still doing a lot of work on this. And so I'm very excited to continue to keep up with that. Some quick acknowledgements. I want to thank the BNZ LTER for housing me this summer. Tamara Harms was my advisor. She was absolutely incredible, super supportive. Jake and Jacob were the grad students that I worked with, same for them. The Jones Lab, another lab in the LTER, helped me establish one of my sites, and the NSF for funding my research. So that's all I got. I'd be happy to answer any questions if you all have any. Yeah.
So, bummer that you weren't able to really compare between like the disturbed streams, like the permafrost and fire, ones that didn't have.
Yeah.
And when you looked at those patterns, were they really just all over the place, or did it look like something might be going on and you just needed a bigger sample size to like, secure the pattern? What did that actually look like?
Yeah, it did seem like the sites that had a lower permafrost extent had more, had less nutrient limitation, which would make sense because, you know, as the permafrost is thawing, it's bringing these nutrients into the streams. But other than that, not really, unfortunately. Like the unburned catchments were not very productive ecosystems anyway. But neither of those were limited by nutrients, most likely because they were limited by like sunlight. So yeah, 2 out of the 3 of the unburned sites were not limited, which doesn't make sense.
So you already answered my next question. If they're not nutrient limited, then what's going on?
Yeah.
Those other streams?
Yeah, most likely sunlight. I'm sure there are some other things that could be going on as well, but yeah. And then I think I have a question in the chat.
We sure do.
Oh, perfect.
I bet you can't guess who it asked. Jim's asking, how do the nutrient levels you tested compare to the levels of nutrient influx that occurred due to post-fire runoff and permafrost melting?
To be honest, I'm not sure. I would love to look into that a little bit. I know my mentor and I kind of had a little bit of conversation about this, but I don't know the degree to which nutrients are actually introduced into these ecosystems after these disturbances. But yeah, that would definitely be an interesting and really insightful thing to look into, I think.
Future research needed.
Yeah, for sure.
Awesome. Well, thanks again, Margaret.
Thank you.
Last but certainly not least, Mary Grace. Grab that menu for you.
So again, you'll probably want to click the first time and then it should work.
Okay,
All right.
thank you. Okay, so this past summer, I was in Morehead City, North Carolina, and I was doing research on blue crabs, specifically their size at maturity. Oh, I have to click. Okay,
There you go.
I got it. Okay, so why is this important? Blue crab fisheries on the East Coast, there's a very high demand for them. They are definitely overharvested and their population numbers have been decreasing. And understanding blue crab size at maturity is important in effectively managing sustainable crab fisheries. So it's important that we keep all of this data up to date so that we can keep policy up to date and effective as well. A fun fact about female blue crabs is they do not molt after reaching their reproductive form. So once they get their first sponge of egg, they don't grow anymore after that, which makes it really easy for me because if I get a crab and sponge, I know that's their size at maturity. There was prior research done by the North Carolina Division of Marine Fisheries. I think the research went from like 1988 to 2016, was published in 2018. And they had 3 different study sites, one at Chesapeake Bay, one in North Carolina, and one in Florida. And they found that size at maturity for blue crabs in the Chesapeake Bay had increased by almost 27 millimeters in only 16 years. And since no research has been done since then, size at maturity data has not been collected for North Carolina blue crabs in over 6 years. And there's many environmental factors that can go into affecting a blue crab size at maturity and reproductive success, and this includes salinity and water temperature. So I had 2 study sites. They were in between Morehead City on your left and Beaufort is like right over here on the right. The top one is called Haystacks and the bottom one is Prythurge Creek. These will not show up if you look them up in a map. I think that's just what locals call them. So I went ahead and added coordinates as well. So for my methods, I collected blue crabs from both locations and I used 3 different kinds of traps or pots. The reason behind this is I wanted to make sure that I was collecting all different sizes of blue crabs. So minnow traps work better for collecting small crabs, and then standard pots and peeler pots will get the medium and larger crabs. Once I collected the crabs, each female was weighed, checked for sponge, and their carapace width was measured. So over here, you can kind of see what a crab sponge will look like. They're really gross to touch, very mushy, almost like mashed potato consistency. So the bright orange will mean that they're early on in their development, and the darker color will mean that they're further on in their development. In June and July, when I was collecting them, they were mostly like bright orange to like very light brown. So basically these 2 rows of colors. From this data, we compiled the individuals by their shell width or their carapace width, and we fit a logistic regression curve to the proportion of mature individuals at each given size to estimate the overall L50 value. So L50 value is what was used in the prior research done by NCDMF, which is, so I just kind of replicated it so I could compare my results. And it's 50% mature individuals and 50% immature individuals at a specific carapace width. So we compared our estimated L50 value to the prior research that was done. So here are my results. On the left, you can see the length distribution. It ranged from 110 millimeters to 180. And the average was in between that 140, 150 mark. And then for weight, there was a big range there, 50 grams to 300 grams, and the average was right under 200. And from that data, I made this graph on the left. This is that L50 value I was talking about. So if you look on the y-axis and you go to the 50 mark and go all the way over, right up here would be our L50 value. And then this, I just used the data that was collected from NCDMF from 1988 to 2016, and I added my value over here on the right. There's 2 issues with these graphs. My confidence interval is huge, and this is very high. So my estimated L50 value was 167 millimeters at 50% maturity for both locations. An increase of 43 millimeters in 6 years is not very likely. And so the error was probably had to do with my sample size. When I was designing this experiment, I was under the impression that I would have a much larger sample size, but life happened and COVID happened and weather happened and there was like a small boat advisory. So I only got 91 total female crabs and only 17 were in sponge. So why is the L50 value so high? We would expect that if I were able to collect more individuals, more of them would have been in the 120 to 130 millimeter range. And this would result in an L50 value much closer to the mean. Looking at the Chesapeake population trend, it is probable that the L50 value had increased a lot, but probably not as much as my results indicate in North Carolina. Acknowledgements. So I was funded by the Doris Duke Conservation Scholars Program, and this was the grad student I was working with, Alex Rocco, and another grad student in his lab, Matt Damiano, helped me with all of the stats stuff. So huge thanks to them. And all of this was done at CMAST in Jay Chow's lab. Does anybody have any questions? I have one. How do you think size selective harvest would affect reproductive size over time? That's a good question. So when it comes to forming policy for harvesting blue crabs, we can try to prevent smaller crabs from getting harvested so that they have the potential to reach reproductive form, especially for female crabs at least. And honestly, a lot of the times when they are in their terminal molt and they have that sponge on them, they won't be harvested because they look kind of gross. So I think understanding size at maturity for female crabs can do a lot when it comes to policy for harvesting them. Any more questions? Oh, yes.
But why are the crabs getting bigger?
Why are they getting bigger? A lot of it has to do with environmental changes. So salinity and water temperature are the two big ones that I found in like literature research or reviews. So with the changing climate, obviously, these are causing changing water conditions. I don't know exactly the science behind it, but I know that those two factors have a big correlation with their size.
I did have a
Mhm.
one. So I like the idea of having giant crabs in the future. Is there perhaps a selection that, so is it easier to spot and find larger crabs? And so kind of with your limited sampling, it might have been biased in that direction. I guess I don't understand how you caught and sampled the crabs, and that might be a reason for bias. It was just easier to catch larger crabs or something.
Mhm.
So.
That is why I used 3 different kinds of traps. So I had minnow traps, which we caught a lot of small crabs, but they just weren't in sponge because they're little babies. But the minnow traps are very small. The entrance is like that big. And then I did have standard traps and peeler pots as well, which allows for those medium and large crabs. The research that Alex was doing was a completely separate project, and he was doing research on growth and mortality rates for crabs. So the whole point of his project was to collect crabs of all different sizes. So I was really lucky to kind of have that advantage. But yeah, we collected smaller and larger crabs. It was just the larger crabs tended to be the ones that had sponge.
And those are the same traps and methods that previous people had used before, so it's not kind of.
They actually are different methods. So his research was centered around the NCDMF research project that was done, but they were using trawling nets and we were using traps, mostly to just do with funding. They have a lot more money than a grad student does. So there could have been some differences there.
Maybe the bigger crabs are just easier lured to the traps or something like that.
Yeah, I honestly don't know. That would be interesting to look into.
I have a question about your methods. You
Yes.
said you used the L50 as a measurement of it because that's what was done before.
Mhm.
If you had your pick, would you have chosen a different metric, a different measurement that
I'm honestly not sure. Stats has never really been my thing, so the summer was the first summer I was really diving into it. I think L50 value was interesting to me because I don't really understand exactly why they did it that way. I don't understand why the other female crabs that weren't in sponge were also added into the mix. And I think since I did have so many that weren't in sponge and then only a few that were in sponge, it just wasn't very equal. But I think if I had a larger sample size and I was able to randomly choose from the ones that weren't in sponge and so that it was an equal proportion, it might have worked out better. But I'm not sure. Awesome. Thank you.
Thank you.
Oh, and Michelle wants to know how a crab earns the title Crab of the Day.
Okay. So it was actually Crab of the Day on Twitter. So we took that picture. That's a male crab there. And my grad student posted it on Twitter, and there's a page that posts crabs every day. So we got Crab of the Day, retweeted June 21st.
Awesome, awesome.
Yeah.
Well, that concludes day one. I want to, let me see if I can pull up our. Maybe, yeah, there you go. I'll remind you, we have day two, 4 more presenters on Friday at noon back here, same bat time, same bat channel. Join us via Zoom if that works better for you. Our presenters will be presenting in person. Huge thanks once again to our 4 presenters and to all of our attendees. So thank you, thank you. Second installment of the Applied Ecology Minors Research Symposium here in fall 2022. I got the year right. Okay, it's one of those semesters. I hope you're all doing really well. Really excited to be back after 5 semesters online, to be back sharing a room space with you all. So thank you for taking time out of your busy Friday. Is it the first reading day? No.
Next week.
Oh, not yet. Okay. So on this busy Friday. Our lineup today, we do have a slight change in our program today when students had to reschedule. Not to worry though, I will email all of the Applied Ecology listservs when we reschedule so folks can see Ian War's talk if you're able. But I'm really excited for us to have talks by Sophie Meng, Teddy Sarian, and Eli Benbenek. Before we get started, I want to acknowledge that we are on the traditional lands of the Skarura Tuscarora, the Catawba, and the Lumbee tribes. The goals of applied ecology are to study the natural, non-human world around us. Often we can apply what we learn to better manage or conserve natural resources. But it is important to note that way before Western science and Western scientists decided that this was important, expert knowledge of the natural world was already applied as an integral part of indigenous cultures. We are privileged to be able to practice science and to study ecology in academic settings, but we do need to remember that the Western, the scientific, the academic way is not the only way, and it's likely not even the best way. Acknowledgement is a form of decolonization, dismantling an existing power structure. In order to change systems of oppression, we need to first acknowledge that the system is built on oppression. North Carolina State University is a land-grant university, which means it originally received funding through the Morrill Act, signed into law by President Lincoln. That was federal legislation that legalized the purchase or forcible taking of land from indigenous tribes. Indigenous lands raised $134,920 to fund NC State. That's the equivalent of $3,810,578 today. I hope that we can disrupt and dismantle colonialism beyond a simple territory acknowledgement by supporting and empowering each of you as scientists. So, without further ado, I'm going to close out this PowerPoint, reopen another PowerPoint so we can welcome Sophie Meng to present. You may need to click for your first move, but then the clicker should work. Okay, all right.
Hello, everyone. Thank you for being here, and thank you, Erin, for organizing all of this. My name is Sophie, and I have been working in the Sheth Evolutionary Ecology Lab for the past 2 semesters with Dr. Seema Sheth and Caleb Goff. My main project has been to create an occurrence data set for plants growing in alpine regions, which is what I'm going to be talking about today. So my project is adjacent to a much larger project that has been going on for a long time all over the world, which is GLORIA, which stands for Global Observation Research Initiative in Alpine Environments. Both of my project advisors have been working with GLORIA specifically in the Great Basin region over the past few years. So that's how I made that connection. But basically there's a team of researchers, botanists, and other volunteers that travel to the mountains in the Western United States to survey the plant community on those mountaintops every summer. And so over the past 19 years, we have compiled a long list of species that have been observed during those surveys. And a lot of those species are very rare and can only be found in those alpine environments, which makes them important indicators for climatic change under global warming. And that brings me to my next point, which is that using these surveys and the data set that I'm creating could be essential to understanding and predicting climate change on mountaintops. Some other important reasons for why we'd want to create such a data set encompassing all the plant occurrences on mountaintops is that we can use their coordinates to estimate climatic niches, which is important for predicting range shifts. And that also includes defining the climatic niches for species that are colonizing more often and increasing in cover, like plants that are warm adapted, which would be known as thermophilization, and also plants that are wet adapted, which is known as mesophilization. Also, having a data set like this is a good base for any study that requires a history of a plant species range or community composition. Finally, we need a good complete data set of alpine plants because it doesn't exist yet, and the data sets that do exist are bound to ecologically irrelevant boundaries like the borders of California, for example. So our goals for this project were to create this all-encompassing data set from the species observed in the GLORIA Great Basin Mountains that includes only verified and likely occurrences. We also want to accomplish the cleanest possible data set to avoid overestimations of species ranges and any skewed analyses. Additionally, we want to be able to visualize and estimate plant ranges for some of the species that my advisors are specifically interested in. And finally, we aim to provide this occurrence data set for various future research projects focusing on alpine plant communities. And this picture is of Androcerae septentrionalis, which is one of the species that PhD student Caleb Goff is interested in. We're actually running a germination trial on that right now. So to accomplish all of this, we used GBIF, which stands for Global Biodiversity Information Facility. And GBIF basically collects all the data from all kinds of sources on plants and compiles it all into one big open access database. So you can imagine that there's a lot to clean up here if you want to use this for publishable research. So to do that, we downloaded all the occurrences for the 227 species on our list from GBIF, and then created a script in R to automatically clean that. So some of the requirements that we had were that we were looking for records newer than from 1948, no records from capitals, seas, institutions, or GBIF headquarters, and none in urban areas as well. And we were also able to remove outliers with our script. So after we'd figured out a smooth way to do this, which took longer than you might imagine, we were able to move on to manual cleaning, which we decided to do in Google Earth Engine because I'd already had previous experience with that, and it seemed the easiest way to do it. So how we do that is we separate out one species at a time in R from our cleaned list and import that into Google Earth Engine. And then we literally zoom into each occurrence that seems suspicious to us and kind of look into it further with GBIF or other resources like iNaturalist. So our results so far are that we originally had around 150,000 observations in our data set that we downloaded from GBIF. And now we have around 37,000 left over. So we cleaned about 110,000 records that were likely inaccurate or too old to use. And then this is a map that shows some of the flagged records that were all removed, but this is actually the minority of all the records that we removed. And then this is a part of the script that we used to clean the data set and flag those records. So on the very bottom line, you can see the tests that we ran. Some of those are that the capitals, we got rid of duplicates, we got rid of anything in institutions, urban areas, and seas. And so this image might be a bit blurry, but hopefully you can see that this is an accumulation of species occurrences, and they're all listed on the right here. This is kind of cool because it shows the overlap between species ranges and also the difference in size of those ranges. And it also makes it kind of clear that a lot of these species are concentrated in one area, and that area is not always California. But like I said, when cleaning, we only look at one species at a time because looking at it like this would be a bit chaotic and would probably take longer. So this is what it looks like when we're actually cleaning the data. And just as an example for what I would look for, these 3 occurrences on the left would immediately be removed because they're in Russia. So I would immediately just write down their GBIF ID so that they can be removed from the data set in R, as we're only looking for North American occurrences. And then the other circled points would also be looked into further because they're kind of farther, they're on the boundaries of the plant range, the species range, and they're they're just a little isolated from the rest. So probably most of those northern occurrences will be looked into as well. So that's pretty much where I'm at right now with this project. It is ongoing, as I have also taken on some other projects, and we have just actually recently, like this semester, started working on the manual cleaning process because, like I said, our script was causing some issues and took a while to work through. But moving forward, once we're done with all the cleaning, this data set could be used for many different things, like I mentioned earlier. For example, looking at latitudinal and altitudinal range shifts and using that to understand species turnover on mountaintops, especially under climate change, which is important when it comes to alpine species, especially because, as some of you probably know, mountaintop extinctions are kind of a threat to a lot of alpine species. And also, like I said, those climatic niche definitions can be helpful when studying a specific ecosystem or species. And this data set could also be used for studies on species relationships or just overall information and education. So here's some key takeaways and kind of improvements that I would take into consideration if I were to do it all over again. First off, I think a lot of code crafting time could have been saved by tightening up the parameters for download from GBIF from beginning on. We kind of had to go back a lot of times and tighten up those parameters several times. And another thing I learned is that taking notes, especially when coding, is very important. Just taking note of all the mistakes you make and what didn't work and what does work. Especially when you're working on a project that's kind of long-term like this one. Something I noticed while manually cleaning is that depending on how you refine your data and what database you're looking at, a plant's range might look different or might appear different. For example, when I'm looking at iNaturalist records for a certain species, there's usually a lot more records altogether than in the clean data set that I'm looking at in Google Earth Engine. But at the same time, a lot of those records that are in the kind of hard to reach places, like very northern records, don't appear on iNaturalist. So that's kind of a difference that I noticed while cleaning. And another thing I've run into is that some records simply have to be removed from the data set because there isn't enough detail, like there wasn't enough written down when they were collected in the field. And so basically, if you ever get the chance to record a collection species, just make sure to take note of as many things as precisely and accurately as possible. So finally, I'd like to thank Caleb Goff, who is my main advisor and inspiration for this project, and Dr. Seema Sheth, who has also been a huge help to this project. And I'd also like to thank Emma Wilson and Matthias Vanderbunk, who are the other undergrads in my lab, who have been helping me with the manual cleaning process. And thank you all for being here. I'd be happy to answer any questions that you have.
I might have missed it, but you did the initial cleaning. Why did you not use anything before the release?
A lot of those are the coordinates are kind of inaccurate just because they weren't using GPS technology back then, and also we had to use some cutoff for just preciseness, I think. Yeah. Yes.
You use some filtering rules, like in the steps as you're going through the automated process. And it looked like you did a ton of manual filtering to remove data points. If you could write one more filtering rule, like if you were to add one to that list, was there a common enough situation where you encountered the same sort of reason for excluding something that would be a useful addition to that list of filters?
Yeah, I think one of the filters, which is the C's filter, is very helpful, but at the same time, I'm still finding a lot of occurrences in lakes or inlets, and so I think that would be a good addition if there's a way to refine that somehow. And then also, I'm not sure if there would be a way to do this, but a lot of the time when I'm looking at records in GBIF, I'm looking for a digital herbarium sheet scan, and so if that's missing, a lot of the time we have to remove the record if there's also no iNaturalist records nearby. So that would be another thing that keeps popping up.
It looks like you've got some questions in the chat, which is of
Yep.
course popped right back up.
Skylar's got one here.
Awesome.
Cool. Hey, Sophie, that was really awesome. And I'm so happy to see you using Google Earth Engine still. That's very exciting. I had a question about like where the INAT records come from. So we've played around with the GBIF package for our little bit for snakes. And even though like GBIF as a whole pulls in the research grade INAT observations, it doesn't look like we get those in the R package. Is that the case, or like, are you pulling in like GBIF and INAT separately and then merging those?
So for our data set, we're not using any iNaturalist occurrences actually.
Yep.
We're
Okay.
only using verified like herbarium.
Gotcha.
occurrences. Yeah.
Okay. But then you've like done some comparison to INAT to see how that would change things potentially, or like look a little bit different.
Right, yeah. We use that to kind of confirm if it's likely that an occurrence would be there.
Cool. And then I was also wondering, like, have you considered trying any filters like in Google Earth Engine? Like, is it worth trying like an elevation filter to, you know, in addition to like the seas filter from GBIF, could you exclude other low-lying areas, or do you think you would lose important points if you do that?
I think that depends on the species you're looking at. It definitely could be helpful for some of them, especially probably the ones that are only found on mountaintops. But we haven't tried that yet, no.
Okay. Cool. Thank you.
Yeah, thank you.
Awesome. Thank you, Sophie.
Thank you.
Up next, we have Teddy Sarian. Spoilers. Let me go ahead and grab that menu for you from the top. This is the magic that will be edited out. All right, and there's a small chance you'll have to click once on the touchpad to get going.
Okay, thank you.
Yeah.
All right. Hello, everybody. My name is Teddy Sarian, and today I'll be talking about the parasites in Radineia flavellata, which I'll be referring to as pinewood snakes for the rest of this presentation. So parasites are a very diverse group of organisms that make their living exploiting their hosts. They're important in ecosystems because they have big impacts on host populations, such as agricultural pests or endangered wildlife species. There are many parasite species that are involved in food webs, and these trophic interactions help us to learn more about the host. For example, many parasite species have complex life cycles that involve trophic transmission, where the host becomes infected after eating prey. So we might be able to learn more about the presence or diet of the host by looking at the parasites that are in the ecosystem. But we don't know much about parasite species. For example, our lab, the Shell Lab, has been doing a systematic literature review of the parasites reported for snakes in the United States and Canada. Out of more than 150 snake species in these countries, only about 50% have ever had their parasites described. This suggests that there are probably many more parasite species out there that we have not yet described. From that review, we discovered that the pinewood snake had no known parasite species. The pinewood snake is native to the southeastern United States. And in North Carolina, it can be found in the southern coastal plain region. It's a pretty small snake, on average being about 1 foot long, and it lives in the pine flatlands and oak forests and eats small frogs and lizards. Since we don't know much about these secretive snakes, the goal of my project was to describe their parasites for the first time. So the pinewood snakes that were used in this survey came from, they were roadkill snakes in a relatively small area of Clinton, North Carolina. And there's a person out there in Clinton that likes to look around for roadkill snakes. And when they find them, they freeze them, send them to a museum. And then that museum donated 15 pinewood snakes for us to dissect. In the lab, we thought out the frozen snakes. We took general data on the snakes, such as length, body conditions, etc., and then opened them with an incision from the head to the cloaca. Most snakes were adults with sex organs, so we know that 2/3 of the snakes were female. We examined every organ using a dissecting microscope to look for parasites by squashing the organs on glass slides. And then as well, we also looked at the mouth and the body cavity once the organs were looked at. If we found parasites, we examined them under a higher level of magnification, and we can already identify all of the parasite types based on morphology, which I'll talk about later in this presentation. We can also try to use anatomy and genetics to identify the parasites down to the species level, which we are still working on. A strong majority of the found parasites were saved in ethanol vials for future genetic analyses. And bad for the snakes, but good for the researcher. We did find parasites inside of these snakes. So we'll be the first study to describe pinewood snakes macroparasites. Most snakes had at least one parasite species with an overall infection prevalence of 64%. A grand total of 210 individual parasites were found in these 15 dissected snakes. We mostly found the parasites in the body cavity, with the stomach and the intestines being the most infected organs. And over 11 times as many parasites were in the body cavity compared to the most infected organ, which was the stomach. And then it is worth noting that since these are roadkill snakes, there were 0 snakes of the 15 that had all of their organs. And the average snake was missing about 1/3 of their organs. I'm confident more parasites could have been found in these 15 snakes, but we were unable to due to body conditions. Right. Also, one notable snake, it had a total of 77 parasites, which is insane. So the first major taxonomic group that we'll look at is the trematodes. We found a total of 193 individual trematode parasites inside the 15 snakes, and most of them were Mesecar, a larval stage that forms as a cyst. These are usually impossible to identify by morphology alone, so we aren't sure how many species we have yet. Because Mesecaria are usually tropically transmitted, finding them in pinewood snakes suggests that pinewood snakes are serving as an intermediate host for these parasites to some downstream predatory hosts. And going back to the 3 cyst walls point over here, this is how we would tell that it's trematode Mesecar. So there's one right here, and then another up here. And then this in the inner one is created by the cyst, but this outer one is created by the snake's immune response. Of the 193 total trematodes received from the 15 pinewood snakes, just under 90% of the trematodes recovered during the dissections were located in the body cavity, not in the organs. The next group we'll look at is the nematodes. We found a total of 15 individual nematode parasites inside of 3 of the 15 snakes, giving us a 20% infection prevalence. A majority of these nematodes were in the J4 stage, which is a larval stage that serves as the predecessor to the adult form. Recovered nematodes were concentrated in just 2 organs, the intestine and the stomach. We only ended up, our next group that we'll look at is acanthocephalin, which we only ended up finding one, and we only found 1 acanthocephalin in 1 snake. And acanthocephalin are easily identified by their spiked proboscis, which they use to attach to their host organ. However, we found ours, the proboscis was inside still of the acanthocephalin. It was not in its adult stage yet. And that one was also just found in the body cavity. So even in a small sample size of pinewood snakes, 15, we found several parasite species from different taxonomic groups. These parasites suggest that pinewood snakes are intermediate hosts for parasites that also infect their predators. We'll continue to work on species level identification, which will likely require genetic analyses for many of the larval parasites. We found a plethora of parasites in these snakes that had never been examined for parasites before. By continuing this study and studying other snake species in the 50% that have never, 50% of snakes that have never been examined for parasites before, we can greatly expand our knowledge on snake parasites and their ecology. And for acknowledgements, I just want to say thank you to Dr. Skylar Hopkins and PhD student Alex Nelson, because this project would not be possible without all of their help and efforts. And feel free to ask any questions that you have about my project, and thank you for attending my presentation.
Can I hop in with a question real quick?
Yeah.
You said 77 parasites was really high. What's a more typical or expected parasite load for a snake?
So it does vary a lot, and since we only had 15, it's not probably not a great estimate for all time with snakes, but I think on average I would come across probably like single digit numbers, but it's hard to say
Wow.
where in that range it would be.
Awesome. Thanks.
And then Kenzie said, would infection by these parasites be asymptomatic in these snakes? I do not know for sure, because I only deal with the roadkill snakes for this project. So yeah, I am not sure if they would be asymptomatic, but it would make sense for them to be.
Great question. Any other questions here?
Mhm.
These snakes were obviously hit by cars, but were they people with parasites? Like, was the one who had 77, was that potentially in the
I'll give my best guess. So I think for the most part, most of the time parasites, they don't want to cause severe sickness in their host, because then the parasite, they won't have a host anymore if the host dies. So typically the snakes will be fine with at least a few parasites, but for a snake with 77, it's hard to say if it would fare well in that condition.
Thanks so much, Teddy. Have you got another question? there?
Yeah, Alex asked, what was my favorite parasite I found? The acanthocephalin, just because it was a unique one.
Awesome. Thanks again. And thanks everybody for your questions. I'm going to transition out. Last but certainly not least, we have Eli Benbenek. Rescue your menu up here, and you may have to click on the trackpad for the first one to advance. All right.
Okay, so hi, everybody. I'm Eli Benbenek. Just a little disclaimer, my voice is being a little, going a little haywire today, so warning in advance. But today I will be talking to you about my project, which was comparing the behavior of wild and captive box turtles. So just to start off with some background information on the eastern box turtle. The eastern box turtle is the state reptile of North Carolina, and they fill a very important niche in their ecosystem. So they eat a wide variety of food, from berries whose seeds they distribute to various species of plants, flowers, other animals, and even scavenge dead organisms. And in addition to that, they also burrow when they hibernate in the winter. So of course, when they're done hibernating, other animals are able to use this. So these behaviors just go to show, even though the eastern box turtle is kind of tiny, it's got a very wide range of roles in its ecosystem and fills a very important niche. And in North Carolina, or just in general, it's listed as vulnerable by the IUCN due to a bunch of anthropogenic factors, some of which are habitat loss, obviously, pet trade collection, and of course, road collisions. So just these are some of the reasons why I focused on the box turtle for this project. And just to get into a little bit of what the literature has to say about box turtle behavior and wild box turtles. So first and foremost, wild box turtles move around to different habitat sites depending on the season and short-term weather conditions. So because they are ectotherms, they kind of have to rely on the environment to regulate their own, or to regulate their temperature, since they cannot do it on their own. And turtle locomotion was found to be strongly affected by temperature. And this is usually as temperature goes up, so does their activity level. But of course, there's probably an optimal range, or there is an optimal range. So if it gets too hot, they have to go into kind of moisture and energy like retention, which kind of goes into the last point. Turtles select their microhabitat microhabitat to minimize water loss and to thermoregulate. And some of these factors, which obviously they probably take into account, are humidity levels, surface temperature, ground temperature, and plant coverage. So they kind of are constantly working to make sure they maintain their optimal temperature and moisture levels. So for my project, my purpose was to compare the behavior between captive turtles and the wild radio transmitter turtles at the Piedmont Wildlife Center. And as I was kind of researching my project topic and what I was going to do, I was looking for previous literature to see if there was any kind of say in if this had been done before, and I did not find anything. So the lack of research on the topic makes it significant enough to look into, and also possible applications of the findings. So if you know anything about turtles, a condition that they're really susceptible to is called a oral abscess. And I didn't include a picture because it's kind of gross, but it's basically just a swelling in its neck area. So, and that's its ear. And the cause isn't clearly known, but it's very prevalent in both captive and wild turtles. And the recent literature has kind of suggested it's due to vitamin A deficiencies, and at least in captive turtles. But in wild turtles, it's also been linked to pesticide exposure. So I think maybe a possible application of a comparative like behavioral study like this could be used to maybe look at the pathology of oral abscesses in wild and captive turtles, just to kind of like look at the pathology, see if maybe some of the causes or effects of the disease could be better explained, like by comparing captive and wild turtles. So to go into the methods of how this data was collected, this was done at the Piedmont Wildlife Center over the course of the summer. And to track the wild turtles, we used the radio receiver right here, and each turtle, as you will later see, is transmitted with a radio transmitter. So you kind of just start the cabin and it'll beep. And as you get closer to it, it'll beep more and more and more. And then eventually you look along the ground for it. And whenever you do find the turtle, it's kind of hard to see, but here's a lot of the data that you're looking for. The behavior, the air temperature, ground temperature, the cloud coverage, and latitude, longitude, and the time of day as well. So to go into the behaviors that were coded, there are only really two behaviors that the turtles were found to be as for outside. So as you can see on the left, this turtle is buried. So that basically means it's under some kind of substrate, whether that be pine straw or branches or logs or even inside of pipes. And obviously, like mentioned before, this is probably just to maintain its thermoregulation to kind of cool down since it was the summertime. And the other behavior is visual, and if it was visual, that basically just means that it was above the ground, not under anything. And this could be outside or inside of the shell, since most of the time, probably when we get close to it, it's going to go back into its shell and kind of chill out. So to go into the indoor turtles, this is a much easier process, but it's very similar. So as you can see on the left, that's the same kind of the visual. It's outside of its enclosure, and it might be walking around or just chilling out. And then on the right, you can see the buried. All of them have the kind of the structure inside of their enclosure that you can see if they can get inside of and kind of cover themselves a little bit with bedding, and that's the closest you can get to an outdoor turtle burying itself. And I went around to the 5 cabin turtles that were inside, and then there were 6 turtles on the outside that had radio transmitters on them. And so by the end of the study, there were 40 wild turtle entries and 45 captive turtle entries to use for comparison for their behaviors. So to get into the results, this is figure 1. And as you can see, the wild turtles are right here on the x-axis, and the captive turtles are here on the x-axis also. And then on the y-axis, you can see the number of behavior occurrences. And here you can really see that the wild turtles were buried much more often than not, and then the captive turtles were visual more often than not. And to kind of better visualize this, I have a pie chart, and here you can see it's actually kind of interesting. The buried has the same count as the visual at 22, but you can also see that the buried was 64% of the time, while the visual was 51% of the time. So you can see that the captive turtles were much more visual and like moving around. So I also tried comparing the behavior to the time of day and kind of seeing if also the wild turtles separately had some kind of relationship between ground temperature, air temperature, cloud cover, and their behaviors, but none of the data I could really get to work, so I kind of left it out of the presentation. And you can also see there's this other that the captive turtles have that the wild turtles don't, and that is because the captive turtles were seen to be feeding and also to be soaking, which we're not seeing in the wild turtles. So here you can kind of see they all have these water containers that some of them like to just hang out in. So that was those. And lastly, my hypothesis was supported, as you can see, the indoor turtles were much more active. So for the discussion, difference between the groups affected or was affected by many uncontrolled variables. And some of these differences could have been the temperature, humidity differences between the outside and the cabin. The cabin temperature, since it was summer, could have been more within their optimal range, whereas the outside would have been much more warm. And that could have caused them to be more buried so that they could, obviously, like I mentioned in the introduction, maintain their internal temperature and their water amount to be at a good level. And of course, there are other differences, such as the difference in diet, maybe the daily activity schedule, since the turtles are more used to, or the captive turtles are more used to people being inside, like hanging around with them. And of course, our interaction could have accounted for some of the changes in behaviors that we're seeing. Also, since there's no prior literature, the results, there isn't really much to compare to, but the results did give a good baseline for another comparative study to follow for future research. But obviously, if that's the case, there are limitations that should be addressed for future study, such as human interference, a small sample size for both the captive and the wild turtles, time disparity, since the times were captured differently for the wild and captive turtles, and it was harder to control a lot of the variables for the outdoor turtles. So a future research project that could kind of capture the essence of this one, but still advance it, could be to use really similar methods, but to observe the turtles on rainy or excessively warm days to explore the role that weather conditions could have on both the captive and wild turtles and kind of the difference between them. And a good way to kind of get around some of the limitations and to have more streamlined data would be to have one person collect all the data for the indoor turtles over the course of the day, and another person collect the data for the outside turtles over the course of the day. And this kind of has the times more streamlined, so you can have the temperature and humidity and all those other variables be more measured and similar, as opposed to this study where it was basically based off convenience, since it was just me going around and doing it. So yes. So acknowledgements. So big thank you to Noelle, and also to Jen and Caitlin for helping me figure out my research projects and make it realistic. And also to the other interns, Tyler and Josa, who helped me collect the data for the turtles. So thank you all for listening. If you have any questions, I'd be happy to answer them. And here's a little baby turtle for you to look at.
Did you notice all that specific individuals of turtles responded doing the same behavior over and over again? Like, do certain individuals tend to hide more?
For the outdoor turtles, I would say that's not the case, but for the outdoor turtles, they all have like 3-letter identifiers. So it's kind of harder to memorize like what their specific behaviors could be. But I know for the indoor turtles, like, I know some of them like to kind of, they have their own kind of personalities. So they have definitely like their own behaviors that I would say are unique. So yeah.
I was curious, what's the range of times that it took from when you turned on the telemeter till you actually found the turtle? Find them?
That's a good question. That's kind of depending on like how close they are and how good we are at finding them. So usually, just to kind of estimate, maybe anywhere from 10 to 15 minutes, depending on how far it is. But of course, once you get up close to it, the transmitter or the receiver will only get you so far where it goes beep, 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 beep. And then it's up to you to look along the ground to see where it is. And of course, if it's like in a log pile, you've got to kind of dig around and be careful not to disturb anything that's might like be there besides the turtle. And if it's buried, like it just kind of adds like, you kind of got to do like a little scavenger hunt puzzle kind of thing.
It's not effective, but I did just want to say that we have added humidity control to our box turtle enclosures, which change for lighting as well. And I've seen almost all of the turtles out way more than they used to be. So,
Gotcha.
and Lynn's spirit hypothesis that they're not hiding because they're a little bit more thermal.
Gotcha. Thanks.
Have you got questions in the chat?
Yeah.
Yeah, 2 questions in the chat. Skylar says, cool project, Eli. How many individual wild turtles did you track, and how long did it usually take to find each turtle?
Oh, gotcha. Okay, so I already answered the time it takes kind of, but there were 6 turtles that had the transmitters on them. So I'm not sure if there was like an even spread for the number of occurrences we had, but I tried my best to do an even spread of like trying to get each 6 like as close to the same number of times as possible. If that answers your question. Yep.
Looks like it does. Kenzie's curious. Many box turtles around here succumb to cars. Do you know of any long-term solutions that would help them cross roads successfully? I was thinking something like a wildlife crossway, but on a smaller scale.
Yeah, that's definitely a good idea. I'm not sure, since turtles are so slow that, you know, that's a hard question to ask. I think a better solution might be going to like the root of the issue. I know that when there's litter on the side of the road, that a lot of animals get attracted to it, and then that kind of goads them into crossing the street when they probably shouldn't be. And I think another solution is just for people to be more aware of their surroundings when they're driving, just so that if they see a turtle, and they're also to add on to that, being knowledgeable that if you see a turtle, maybe you see it heading in that direction, maybe just make sure it crosses the street on its own. If that answers your question.
I was curious if you're interested in comparing these turtle behaviors across different seasons and like what you might predict in different seasons or with climate change.
Yeah.
What does that mean for the box turtle?
I know that the turtles, I haven't been there for the winter, but I know that they get much more inactive and they like hibernate under, like it's easier to find them since they mostly stay in the same spot and kind of get under their brush or whatever they get under. But yeah, it would be really cool to kind of do the same study over different seasons and see how it reacts, because like spring and fall would be really interesting to see how the changing seasons, like maybe they go down and then back up, kind of like a little wave. But yeah. What was the second part of your question?
Climate
Sorry. Yeah,
change.
climate change.
Yeah.
Yeah, that's I'm not sure, because I know turtles are really affected by it, and they're or at least sea turtles are in the reproduction, which is like unrelated. But I feel like maybe they would be more active, like generally over time, like an upward trend up until it kind of passes their optimal range. And at that point, they'd probably like be less active when it gets really hot out, since they'd have to thermoregulate better.
Just constant burial.
Yeah.
Yeah. Really interesting. Thank you so much, Eli.
Yeah.
And thank you all for coming, for attending, for supporting. Thank you, students, for your research. Thank you, attendees, for your questions. This concludes our second installment of the Fall 22 Applied Ecology Minors Research Symposium. Y'all have a great day. Happy Friday. Happy end of semester.